All right, let's dive into something pretty fascinating today.
Yeah.
It's called plasma DNA fragmentomics.
Oh.
And it sounds a little bit like science fiction, but it's really cutting-edge research about these tiny pieces of DNA that are just floating around in our blood.
Right.
And get this.
Oh, okay.
Scientists are figuring out how to use these fragments to detect cancer, monitor organ transplants, and even to get information about pregnancies.
It is really remarkable what we can learn from just these little tiny pieces of genetic material.
Yeah, like what is plasma DNA exactly?
So imagine our cells are constantly shedding these little bits of DNA into our bloodstream.
Okay.
And that's plasma DNA.
Interesting.
And the really interesting part is that these fragments,
Yeah.
they aren't just random bits and pieces.
That's what I was kind of curious about. Like,
Yeah.
what makes these fragments so special?
It all comes down to something called plasma DNA end motifs.
Okay.
Basically, these are specific sequences of nucleotides, the building blocks of DNA, found at the very ends of these fragmented strands. They're like fingerprints that can tell us where the DNA came from and what might be happening in the cell it originated from.
So these end motifs are like clues hidden at the edges of these DNA pieces. What kind of information can scientists actually get from them?
Well, researchers have been studying these end motifs in people with liver cancer.
Okay.
And they've discovered something remarkable.
Right.
A specific motif called CCCA is much less common in their blood.
Interesting.
It's like a red flag waving in the bloodstream, potentially signaling the presence of cancer.
So just by analyzing these tiny pieces, looking for specific patterns, we might be able to detect cancer earlier.
Exactly.
That's incredible.
Yeah.
And this isn't just for liver cancer, is it?
You're catching on quick. This pattern of altered end motifs isn't limited to liver cancer. Researchers are finding similar patterns in other types of cancer too, like colorectal cancer and lung cancer, suggesting that this could be like a really powerful tool for cancer detection in general.
That's mind-blowing. What makes this method so different from other ways of finding cancer? Like biopsies.
Right, scans.
Traditional methods often look for physical changes in tissues or organs.
Yeah.
But end motif analysis looks at something completely different.
Okay.
The actual structure of the DNA fragments themselves. It's like adding a whole new dimension to our understanding of cancer.
Right.
By analyzing the shape of these tiny DNA pieces.
So instead of waiting for a tumor to grow large enough to be detected on a scan, we might be able to catch it much earlier just by analyzing these patterns in the blood.
Exactly.
Wow.
And that early detection could be game-changing for treatment outcomes.
Yeah, for sure.
But what's even more fascinating is that this technology isn't limited to cancer. Okay, I'm officially hooked.
Yeah.
What else can these tiny fragments reveal?
Well, for example, think about organ transplantation.
Okay.
When someone receives a new organ, their blood contains a mix of their own DNA and DNA from the donor organ.
Right, because the donated organ is made up of cells that have their own unique DNA.
Exactly.
But how can scientists distinguish between the recipient's DNA and the donor's DNA just by looking at these tiny pieces?
That's where the end motifs come in again.
Cool.
Researchers have found that different tissues and organs have unique end motif profiles. Kind of like unique fingerprints.
Okay.
So by analyzing the end motifs in the recipient's blood, they can actually pinpoint which DNA fragments came from the transplanted organ.
Oh, wow.
And which came from the recipient's own body.
So it's like having a molecular GPS.
Yeah, exactly.
That can track down the origin of each piece of DNA.
Right.
What does that mean for someone who's had an organ transplant?
It opens up some incredibly exciting possibilities for monitoring the health of the transplanted organ.
Okay.
Instead of relying on invasive biopsies or imaging scans, doctors might one day be able to monitor the organ's function just by analyzing these end motifs in the recipient's blood.
That's amazing.
Yeah.
So just a simple blood test could tell us if the organ is being rejected.
Right.
Or if there are any signs of damage.
Exactly.
That would be revolutionary.
Yeah.
And I'm guessing this same technology can be applied to pregnancy as well.
I got it.
Since there's fetal DNA circulating in the mother's blood.
Just like with organ transplants, researchers have found unique end motif patterns associated with fetal DNA.
Okay.
This means we might one day be able to get a much clearer picture of fetal development and potentially even screen for genetic abnormalities, all through a non-invasive blood test.
That's incredible.
Yeah.
This is really mind-blowing stuff. We're talking about like a potential paradigm shift in how we approach healthcare.
Yeah.
All thanks to these tiny fragments of DNA floating in our blood.
Right.
But before we get too carried away with all the possibilities, I want to make sure we're grounding this in reality.
Yeah.
This is still very early stage research.
But that's an important point.
Yeah.
While these initial findings are incredibly promising, we need larger studies to confirm these results and really understand how these end motif patterns vary between individuals, different disease states, and even different stages of disease.
So there's still a lot more to learn before we see this technology being used in everyday clinical practice.
Absolutely.
But the research is moving at an incredible pace.
It is, yeah.
And I think it's safe to say that we're just scratching the surface of what plasma DNA fragmentomics can tell us about our health.
I agree.
This is definitely a field to watch.
Yeah.
All right. Let's pause there for a moment, and we'll recap what we've learned so far about these fascinating DNA fragments.
Sounds good. So we've talked about how scientists can use these end motifs like fingerprints to identify where DNA comes from.
Right.
But the research goes even deeper than that.
Okay.
They're also looking at the frequency and distribution of different end motifs within a blood sample.
Okay, so it's not just about the presence or absence of specific motifs, but also how often they appear.
Right.
And how those patterns might differ in different situations.
Precisely. And what they've found is that the diversity of these end motifs, the range of different fingerprints we see, is actually much higher in people with cancer.
Interesting.
It's like the cancer cells are producing a more complex or maybe even chaotic mix of DNA fragments compared to healthy cells.
That's fascinating.
Yeah.
So the DNA fragments from cancer cells are almost telling a different story, just based on the variety of end motifs present.
That's a great way to think about it. And this increased diversity of end motifs could be another important clue for early cancer detection.
So we've got these two key findings.
Yeah.
Specific end motifs that are less common in cancer patients.
Right.
And an overall increase in the diversity of end motifs in those same patients.
Exactly.
It's like having two separate pieces of evidence pointing towards the same conclusion.
Exactly. And these findings really highlight how important it is to look at DNA fragmentation in new ways. It's giving us a whole new perspective on the biology of cancer.
Right.
And potentially many other diseases as well.
That begs the question,
yeah,
why are these patterns different in cancer cells?
Right.
It's just random? Or is there something more going on?
That's a great question, and it's something that researchers are actively trying to understand. One theory is that the way DNA is broken down and packaged inside a cell is different in cancer cells compared to healthy cells.
That makes sense. Cancer cells are notorious for their uncontrolled growth and division, so it wouldn't be surprising if their internal processes, including how they handle their DNA, were a bit different.
Precisely. And there's one particular enzyme called DNASE1L3 that researchers think plays a key role in this process.
Okay, back up for a second. Remind me what enzymes do again.
Sure. Enzymes are like tiny machines inside our cells that speed up chemical reactions.
Right.
They're involved in all sorts of processes, from digesting our food to copying our DNA.
Right. Of course.
Yeah.
So what does this DNASE1L3 enzyme have to do with plasma DNA and those end motifs we've been talking about?
Well, research suggests that DNASE1L3 is specifically involved in chopping up DNA, creating those fragments that end up in our bloodstream. And what's interesting is that in the case of liver cancer, they found that this enzyme is much less active in tumor tissues compared to healthy liver tissue.
So if this enzyme is responsible for breaking down DNA and it's not working as well in cancer cells, could that explain why we see these altered end motif patterns in cancer patients?
That's exactly what researchers are thinking. It's like having a pair of scissors that aren't as sharp. So they cut the DNA in slightly different places, leading to those unique end motifs and the increased diversity we see in cancer patients.
It's incredible how these tiny molecular differences can tell such a big story.
I know, right?
It's like a detective novel playing out at the microscopic level.
I love that analogy. And you know, one of the most exciting things about this research is that it goes way beyond cancer detection.
Okay, I'm all ears. Tell me more about how this technology could revolutionize those areas.
Well, we've talked about how different tissues have unique end motif profiles, almost like fingerprints.
Right.
And researchers have taken advantage of this to develop incredible ways to monitor organ transplants and pregnancies.
Okay, walk me through how that works.
Let's start with organ transplantation.
Okay.
Remember, when someone receives a new organ, there's a mix of their own DNA and DNA from the donor organs circulating in their blood.
Right, and we've talked about how scientists can use end motifs to differentiate between the two.
Exactly. But what's even more incredible is that they can actually track the health of the transplanted organ just by analyzing the donor's DNA fragments in the recipient's blood.
So instead of doing an invasive biopsy to check on the organ, they could potentially just do a blood test and look at these end motifs.
That's the idea. By analyzing the patterns of the donor's DNA fragments, doctors could detect signs of organ rejection or damage much earlier than with current methods.
Wow.
It's a complete game changer for transplant medicine.
That's amazing. And what about pregnancy? How can this technology be applied there?
It's similar to organ transplantation in that there's foreign DNA present,
right.
In this case, fetal DNA from the placenta.
Right.
By analyzing the end motifs of these fetal DNA fragments, doctors could potentially get a much clearer picture of fetal development than we can with current prenatal testing methods.
So we're talking about potentially being able to detect developmental issues or genetic abnormalities much earlier and less invasively than ever before.
Exactly. This technology could completely revolutionize prenatal care.
Wow.
And give parents and doctors much more information to make informed decisions.
This is all so exciting.
Yeah.
But I know with any new technology, there are always challenges and limitations.
Of course.
What are some of the hurdles researchers still need to overcome with this end motif analysis?
Well, one of the biggest challenges is that this is still relatively new research.
Right.
We need larger studies with more diverse populations to validate these initial findings and really understand the nuances of how end motif patterns can vary between individuals.
Okay.
We need to be sure we're interpreting the results accurately and not jumping to conclusions based on limited data.
That's a really important point. We don't want to get ahead of ourselves and overpromise what this technology can do.
Absolutely. And another challenge is developing standardized methods for analyzing and interpreting all this data.
We need to make sure that the results are reliable and consistent across different labs and different patient populations.
So it's not just about the science itself, but also about developing the tools and protocols to make this technology usable in real-world clinical settings.
Exactly. It's a complex process, but the potential benefits are so enormous that it's absolutely worth pursuing.
Yeah.
And, you know, one of the most fascinating aspects of this research is the potential to detect diseases even earlier than we can now. Maybe even before symptoms appear.
Now that's getting into some truly groundbreaking territory.
Yeah.
Tell me more about how that might be possible.
Think about diseases like cancer, heart disease, or even Alzheimer's, which are often diagnosed after they've already progressed significantly.
Yeah.
What if we could detect them years earlier when they're still in their earliest stages?
Right.
That could dramatically change how we approach treatment and prevention.
It sounds like something out of a science fiction movie, but it's becoming more and more realistic as this research progresses. It really is. Imagine a world where we could screen for these diseases with a simple blood test long before any symptoms appear.
Wow.
That could potentially save countless lives.
But of course, there are always ethical considerations with any new technology.
Of course.
Especially when it comes to predicting someone's future health.
Absolutely. We need to have open and honest conversations about the implications of knowing our disease risk so far in advance, both for individuals and for society as a whole.
It's a reminder that even the most incredible scientific advancements need to be approached thoughtfully.
Right.
And with careful consideration of the human impact.
Couldn't agree more. But let's shift gears for a moment and talk about something else that's really remarkable about this study.
Okay.
They were able to validate these end motif findings using two completely different sequencing methods.
Oh, interesting. What were those methods and why is that significant?
They used traditional DNA sequencing, which is the standard way of reading the genetic code.
Yeah.
And bisulfite sequencing, which is a more specialized technique that analyzes DNA methylation patterns.
And remind me, what are DNA methylation patterns and why are they important?
DNA methylation is like a set of switches that can turn genes on or off.
Okay.
It plays a crucial role in regulating how our genes are expressed.
Right.
And it can be altered in various diseases, including cancer.
Okay, so they use these two different sequencing techniques to analyze the same samples of plasmid DNA. What did they find?
The really exciting part is that even after treating the DNA with bisulfite, which can be a pretty harsh chemical process, they were still able to detect those distinct end motif patterns.
So it's like those corn edges we were talking about earlier are so unique and robust that they can withstand even intense chemical treatments.
Exactly. And that speaks to the potential of this technology to be integrated into existing clinical workflows.
Okay.
It's not about replacing current methods, but about adding another layer of information, another tool in the diagnostic toolbox.
It's like having a multifaceted view of the same puzzle.
Yes.
Each technique provides a different piece, and together they create a more complete picture.
Exactly. That's the beauty of scientific progress.
Yeah.
It's all about building on previous knowledge and developing new tools that allow us to see things from different angles.
And in this case, those different angles are revealing incredible insights into the world of DNA fragmentation and what those patterns can tell us about our health.
Absolutely. As we continue to refine these techniques and explore new applications, who knows what other mysteries we'll uncover.
Okay, I'm ready to keep diving deeper. But before we move on, let's take a moment to recap the key takeaways of what we've learned so far. All right, so let's recap a little bit about what we've learned about these amazing little DNA fragments and what they can tell us.
Yeah, so we started off by talking about plasma DNA.
Right.
These tiny bits of DNA that are constantly being shed from our cells and just kind of floating around in our bloodstream. And what's really fascinating is that these fragments aren't just random pieces of genetic material. They actually hold valuable clues about where they came from and what's happening in our bodies.
And the key to kind of unlocking those clues lies in those plasma DNA end motifs.
Right.
Those specific sequences of nucleotides at the very ends of these fragments.
Exactly.
They're like tiny little fingerprints that can reveal a wealth of information.
It's pretty amazing what we can find out just from those little tiny pieces.
Yeah, so we talked about how scientists are using these end motifs to detect cancer earlier by identifying specific patterns that are less common in cancer patients.
Right.
And we also discussed how the overall diversity of these end motifs, like the range of different fingerprints we see, is often higher in people with cancer, which would be another important biomarker.
Exactly.
And we even got into like the nitty-gritty of how an enzyme called DNAS E1L3 might be involved in creating those unique end motif patterns in cancer cells.
Right.
Like, it's really a detective story playing out at the molecular level.
I know, it's pretty fascinating.
Yeah. And beyond cancer, we explored how this technology could revolutionize organ transplantation and prenatal care by analyzing the end motifs of donor DNA in transplant recipients.
Right.
Or fetal DNA in pregnant women.
Exactly.
Doctors could potentially monitor organ health and fetal development much more effectively and less invasively than we can today.
Yeah, for sure.
I mean, the possibilities are truly mind-boggling.
Yeah, it's pretty exciting stuff.
But we also talked about the importance of being realistic.
Right.
And acknowledging that this is still very early stage research.
Of course.
Larger studies are needed to confirm these findings and ensure that this technology is accurate and reliable before it can be widely used in clinical settings.
We want to make sure that we're using it responsibly and that it's actually beneficial for patients.
Absolutely. We don't want to get ahead of ourselves and overpromise what this technology can do.
Right.
But at the same time, the potential benefits are so immense that it's definitely worth pursuing with rigorous scientific investigation and thoughtful ethical consideration.
For sure.
I mean, it's incredible to think that these tiny pieces of DNA, once thought of as just mere cellular debris, could hold the key to unlocking so many mysteries about our health. And potentially revolutionizing the way we approach medicine.
I think it really highlights the power of scientific curiosity and the incredible advancements we can make when we take the time to really understand even the smallest details of our biology.
Absolutely.
Who knows what other groundbreaking discoveries await us as we continue to delve deeper into this world of plasma DNA fragmentomics.
This has been a really fascinating deep dive into the world of plasma DNA.
Yeah, thanks for having me.
I don't know about you, but I'm feeling incredibly optimistic about the future of healthcare.
Me too.
Yeah.
It's an exciting time to be following these advancements, and I can't wait to see what the next breakthroughs will bring.
Thanks for taking this deep dive with us. Until next time, stay curious and keep exploring.